Good day everyone. So let me introduce myself for the second time around. I am Miss Girly Biarangas, a senior student here in Bohol Island State University, Calapi campus, taking a Bachelor of Secondary Education, major in English 4. And for today, I will be presenting to you this subject, Science, Technology, and Society, with the title, Gene Therapy. Now, are you ready? Okay, now let's go. Now, as an overview of our lesson, which entitled Gene Therapy, what do you think is a gene therapy? So when we talk about gene therapy, it involves altering the genes inside your body cells in an effort to treat or stop disease. Genes contains your DNA, the code that controls much of your body's form and function, from making you grow taller to regulating your body systems. Genes that don't work properly can cause disease. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and of course, AIDS. Now your next question is, why it is done? Gene therapy is used to correct defective genes in order to cure a disease or help your body better fight disease. Researchers are investigating several ways to do this, including the following: replacing mutated genes. Some cells become diseased because certain genes work incorrectly or no longer work at all. So replacing the defective genes may help treat certain diseases. Next is fixing mutated genes. Mutated genes that cause disease could be turned off so that they no longer promote disease. Or healthy genes that help prevent disease could be turned on so that they could inhibit the disease. And lastly, making diseased cells more evident to the immune system. In some cases, your immune system doesn't attack diseased cells because it doesn't recognize them as intruders. Doctors could use gene therapy to train your immune system to recognize the cells that are a threat. And now you will ask another question. What are the risks in using gene therapy? Gene therapy has some potential risks, of course. A gene can't easily be inserted directly into your cells. Rather, it is usually has to be delivered using a carrier, or called a vector. The most common gene therapy vectors are viruses because they can recognize certain cells and carry genetic material into the cell's genes. Researchers remove the original disease-causing genes from the viruses, replacing them with the genes needed to stop disease. So these techniques presents the following risk. First, we have, of course, the unwanted immune system reaction. Your body's immune system may see the newly introduced viruses as intruders and attack them. They may cause inflammation and, in severe cases, organ failure. Next, targeting the wrong cells. Because viruses can affect more than one type of cells, it's possible that the altered viruses may infect additional cells, not just the targeted cells containing mutated genes. If this happens, healthy cells may be damaged, causing other illness or diseases such as cancer. Next is infection caused by the virus. It's possible that once introduced into the body, the viruses may recover their original ability to cause disease. And lastly, possibility of causing a tumor. If the new genes get inserted in the wrong spot in your DNA, there is a chance that the insertion might lead to tumor formation. Now, what you can expect. Currently, the only way for you to receive gene therapy is to participate in a clinical trial. Clinical trials are research studies that help doctors determine whether a gene therapy approach is safe for people. They also help doctors understand the effects of gene therapy on the body. Your specific procedure will depend on the disease you have and the type of gene therapy being used. For example, in one type of gene therapy, you may have blood drawn or you may need bone marrow removed from your hip bone with a large needle. Then, in a lab, cells from the blood or bone marrow are exposed to a virus or another type of vector that contains the desired genetic material. Once the vector has entered the cells in the lab, those cells are injected back into your body, into a vein or into tissue, where your cells take up the vector along with the altered genes. Viruses are not the only vectors that can be used to carry altered genes in your body cells. Other vectors being studied in clinical trials include, first we have stem cells. Stem cells are the cells from which all other cells in your body are created. For gene therapy, stem cells can be trained in a lab to become cells that can fight or help fight disease. Next is liposomes. These fatty particles have the ability to carry the new therapeutic genes to the target cells and pass the genes into your cell's DNA. Since we're done defining what a gene therapy is, we also know what are the risks and why it is done. Now let's go to the results. So, the possibilities of gene therapy hold much promise. Clinical trials of gene therapy in people have shown some success in treating certain diseases such as severe combined immune deficiency, hemophilia, blindness caused by retinitis pigmentosa, and leukemia. But several significant barriers stand in the way of gene therapy becoming a reliable form of treatment, including finding a reliable way to get genetic material into cells, targeting the correct cells, reducing the risk of side effects. Gene therapy continues to be a very important and active area of research aimed at developing new effective treatments for a variety of diseases. Now, next is heredity. So, heredity is the passing on of traits from parents to their offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parents. Through heredity, variations between individuals can accumulate and cause species to evolve by natural selection. The study of heredity is biology of genetics. The expression of genetic conditions is not always as simple as the amount of DNA present, or the inheritance patterns of a single gene disorder. There are many factors that determine how one's genotype or genetic makeup correlates to one's phenotype or physical manifestations. Among these are penetrance, expressivity, mosaicism, and X inactivation. So when we talk about penetrance, it is a measure of how often a disease genotype correlates to the disease phenotype. For example, in a completely penetrant disease, every individual who is genetically positive for the disease would have clinical manifestation of said disease. In contrast, the diseases with incomplete penetrance, some individuals who are genetically positive for that disease may be entirely asymptomatic. Next is organisms. Inherit genetic information in a variety of ways that result in continuity of structure and function between parents and offspring. Suppose you as a child have a genetic disease, one that could be cured by diet or a similarly straightforward therapy. Now, what are the rules of genes in heredity? So the heredity information is contained or within the genes located in the chromosomes of each cell. An inherited trait can be determined by one or by many genes, and a single gene can influence more than one trait. A human cell contains many thousands of different genes in the nucleus. Heredity factors known as genes are thought to exist as discrete portions known as loci or chromosomes, or of chromosomes. The term discrete refers to the concept that genes are always located at the same point or locus on a chromosome. It is believed that pairs of homologous chromosomes contain linear, matching arrangements of genes exerting parallel control over the same traits. Pairs of genes that exercise such as parallel control over the same traits are known as alleles, which are of two kinds. We have dominant and recessive. So genetic characteristics can be extremely complex and may require the actions of several separate genes to be expressed in the adult organism. An example of trait that require more than a single pair of genes to be expressed are human height and human color. Other traits may be controlled by a single pair of genes. Some of these traits may have extremely phenotypes such as albinism, or which means lack of skin pigment in humans. So there are also common genetic disorder. First, we have the Down syndrome, sickle cell disease, phenylketonuria or PKU, and hemophilia. So each of our gene codes contains unique protein, and each protein has a separate and distinct function to perform in the cell. Moreover, there are also effects of genes in our environment. So genes are inherited, but their expression can be modified by interactions with the environment. Genetic traits are determined largely through the precise information found in the cell's gene structure. A variety of factors in the environment can actually alter the effects of a particular gene. So some example of this effect are as follows. First, we have the effect of light or chlorophyll production. Although most plants have the genetic ability to produce chlorophyll, they will do this only in the presence of light. Without light, these plants produce only a light yellow pigment and therefore appear pale and sickly until they are exposed to sunlight. After a few days of exposure to sunlight, the chlorophyll production mechanism is enabled and green color returns. Next is gene mutations. Genes are segments of DNA molecules. Any alternation of the DNA sequence is a mutation. Usually, an individual cell's altered genes will be passed on to every cell that develops from it. Gene mutations may be defined as being any changes in the nitrogenous base sequence of a molecule DNA. When the base sequence of DNA is altered, the amino acid sequence of the polypeptide for which it codes will likewise be altered. Such an alternation may affect the operation of the resulting enzyme, preventing it from properly catalyzing its reaction and thus preventing a trait from being expressed by the cell. The majority of gene mutations are harmful because they result in the cell being impaired from performing some specific task. In rare cases, a mutation may result in a lethal gene that kills the cell either by producing a substance toxic to the cell or by failing to produce a protein of vital importance to the cell. Gene mutations are passed to every cell that arises from the mutated. If the mutation occurs in somatic or body tissues, its defects is limited to the tissues immediately surrounding the mutated cells. So if the mutation occurs in a primary sex cell, it passed on the offspring that result from fertilization or by gametes produced from the primary sex cell. A mutation can enter the gene pool of a population and be passed on the succeeding generations. This is known to be a cause of variation in a species. DNA exists in hundreds of thousands, if not millions, of different forms, depending on the precise arrangement of nucleotides in the molecule. Its variability is DNA nucleotides themselves are quite complex, being composed of 3 separate subunits. First, we have phosphate group, a chemical group made up of phosphorus and oxygen. Deoxyribose, a 5-carbon sugar made up of carbon, oxygen, and hydrogen. Nitrogenous base, a chemical unit composed of carbon, oxygen, hydrogen, and nitrogen. Bases found in DNA are adenine, thymine. Have you ever written a secret message to one of your friends? If so, you have used a code to keep the message hidden. For instance, you may have replaced the letters of the word with numbers or separable, following a particular set of rules. In order for your friends to understand the message, they would need to know the code and apply the same set of rules in reverse to decode it. Decoding messages is also a key step in gene expression, in which information from a gene is read out to build a protein. Genetic code allows DNA and RNA sequences to be decoded into amino acids of a protein. Now let's have DNA code. The chemical and structural properties of DNA are the basis for how the genetic information that underlies heredity is both encoded in genes as a string of molecular bases and replicated by means of template. As we recall, the DNA is a complex organic molecule composed of thousands of repeating nucleotide molecules, and that each free nucleotide carries with it one of our nitrogenous bases. The particular sequence of nitrogenous bases adenine, thymine, cytosine, and guanine, A, T, C, and G, comprise a strand of DNA and providing the type of chemical code that is understood by the chemical mechanisms of the cell. The DNA code is used by these mechanisms to manufacture specific enzymes and other proteins through the process of protein synthesis. In conclusion, DNA is not merely a molecule with a pattern. It is a code, a language, and an information storage. An essential question to be asked is, how does inheritance help ensure continuity of life? A significant principle of biology is the concept that all living organisms possess a set of instructions in the form of genes that determine the characteristic of the organism. These instructions are unique to the species, for they determine the general physical and biochemical traits of the group. Being unique to the individual, coding of the specific set of characteristics that sets one individual organism apart from all others of that species. Students should also be familiar with mechanisms by which genetic traits are passed from generations to generation to maintain genetic continuity of the species. Molecular basis of genetics that is used to maintain genetic continuity mechanisms by which it changes through recombination, mutation, and genetic engineering. Students should be familiar with the mechanism by which DNA replicate its structure during normal cell operation and in the process of reproduction. The rules of DNA and RNA is the coding of cell-specific proteins and understand the role of these structural and functional proteins, enzymes, hormones, and other substances. So let's go with the genetic code. Every organism requires a set of coded instructions of specifying its traits. For offspring to resemble their parents, information must be reliably transferred from one generation to the next. Heredity is the passage of these instructions from one generation to another. Discoveries in genetic science. The DNA was revealed to be the chemically active agent of the gene. DNA replicates itself when chromosomes replicate in the early stages of cell division. DNA is passed from generation to generation during reproduction and acts as genetic factors. DNA reacts with the cell's chemical factory and produces the observable effects of the phenotype when genes are inherited by a cell or an organism. DNA regulates the reproduction or the production of enzymes in the cell and thereby enables the cell to perform the complex cellular chemical reactions necessary to sustain life.